Okay. Muscle physiology. Yesterday we did nerve physiology. Muscle is one of the type of tissue, the four types of tissue that we know. We have got skeletal muscle, cardiac, smooth muscle. You can see how they are. For skeletal muscle, it is striated. That is why you can see these different lines, striations. And then the nucleus is placed on the periphery. And then it is also not nuclear, just one cell with a lot of nuclei. While cardiac muscle is one characteristic feature that you'll be able to see is that it has got branches. It has branches and also gap junctions there, which you can see, which are known as intercalated discs. Smooth muscles are pieceform in appearance. They are longer, they are more wide on the middle and elongated at the ends. So comparisons of these different types of muscles: a skeletal muscle is multinucleated, a cardiac muscle is branched. Okay, it's branched and it's going to have single cells. Same with a smooth muscle, which is single celled. The shape, skeletal and cardiac are cylindrical, while the smooth is fusiform. Striations are not there in smooth muscles but present in other types of muscles. The nucleus is peripheral for skeletal and central for these others. T-tubules, skeletal muscles, they are going to be there, and they are going to be a center of what is called a triad. They are going to be found on the junction where you have got the A band and I band. While in cardiac muscles, the T-tubules are also going to be there, but they are going to be in what is known as a dyad. There is no triad. While smooth muscles do not have T-tubules, instead they have got a calveoli. These are just depressions in smooth muscles. Sarcoplasmic reticulum is well developed in skeletal muscles. Cardiac muscles less developed. And then for smooth muscles, they are very much irregular. Okay. So those are some things you should be able to look at. And then also regenerative capacity. Skeletal muscles can do they have got limited regenerations. Only say they have got satellite cells which are going to be involved in this regeneration. Cardiac muscles almost don't regenerate, while smooth muscles are very good at regeneration because they can undergo mitosis. Response to load: skeletal muscles can well respond to load, cardiac muscles less, smooth muscles almost do not respond to load. And examples for skeletal, we have got diaphragm, almost all the muscles in the body that you know. Cardiac is just a heart, smooth muscles, the blood vessels, the GIT, the bladder, and other organs. Starting skeletal muscles. Skeletal muscles are going to be lined by an epimecium, and then you are going to have an epimecium and then endomecium. You use EPE. Endo is always supposed to be inside. So when you get a muscle, the entire muscle, skeletal muscle, is lined by epimecium. Muscles are made up of fascicles. Each fascicle is lined by perimycium, and then each perimycium is made up of individual muscle fibers, and those muscle fibers are lined by endomycium. Each individual muscle fiber is made up of myofibril, which is made up of actin and myosin. And that's what you can see making composed of different sarcomeres. Skeletal muscles will develop from myoblasts, which will become the skeletal muscles. Other myoblasts will differentiate and become satellite cells, which can be used to grow in case of anything that happens to the muscle for repair. Organization of a muscle: the skeletal muscles are going to have a sarcomere. A sarcomere is just extending between two Z discs. And then you have a microfibril. A microfibril is just a repetition of sarcomeres. One sarcomere plus another one plus other, and just like that forms a myofibril. These myofibrils are going to have dark ends and light ends. The dark ends are called the A bands. A means the anisotropic or bifringent, and then meaning that they do alter. Okay, they change plane polarized light. They are anisotropic. They change plane polarized light, and then the light bands are the I bands, which meaning isotropic, meaning that they are going to not alter. Okay, they do not change the way plane polarized light to be able to move. So you can see this is it. The distance between two Z discs, that's a sarcomere. The entire length is a myofibril. The A band is made up of both actin and myosin filaments. The actin makes up the thin filament, the myosin the thick filaments. The I band is only having thin filament. You also have the H band, which is the middle. So look at this. This is a Z line. This is a Z line. This is a sarcomere. And then you are going to have the middle of this sarcomere is going to be covered by myosin, mostly myosin. Okay, that's a middle, which is also the A band having the myosin. And then you are going to have now between from the Z line up to somewhere here where it's dark, you are going to have myosin, and that's the I band. That's the actually the I band will just end here. So let me use a diagram that is much okay. This one, you can see that the A band is ending where myosin is ending, and actually myosin is also overlapping with actin. That still makes up the A band. But I band will not include the where the myosin is, just where the actin is ending. What holds the myosin to the Z disc is a protein titin. This is the one. The middle of the A band is the M line. The region of the A band, which only has myosin without actin, is the H zone. Okay. Skeletal muscles, you have titin is to hold the myosin to the Z disc. Okay. So you also have this the rough endoplasmic reticulum in muscle is called sarcoplasmic reticulum. This is the actually a smooth endoplasmic reticulum, not rough. Smooth endoplasmic reticulum is known as sarcoplasmic reticulum, and it is involved in releasing calcium. Depolarization of this SR, sarcoplasmic reticulum, is going to cause the release of calcium, meaning that depolarization, you know that it is the increase of positive charge inside the cell. So if action potential enters the T tubules, it is going to cause the sarcoplasmic reticulum to be able to release calcium. To trigger calcium release from sarcoplasmic reticulum, the fiber simultaneously okay, so what is happening is to cause the release of calcium and also contraction of the myofibril. Sarcolemma is going to be folded and is going to have T tubules. So T tubules are going to be found between A and I bands, and that is where action potential will be able to flow. Now, the mechanism of contraction of skeletal muscle. During contraction, you need to know that the thin filaments and the thick filaments do not change their length. The only thing that is happening is that there is an increase in overlapping between the thin filaments and the thick filaments. And so these thin and thick filaments are sliding over past each other, not that they are reducing in length. Contraction is going to be induced when an action potential arrives at a synapse. Yesterday we talked about nerves. A synapse is just a joining of a nerve and a muscle. At that point where they join, there's a synapse, which is known as a neuromuscular junction. Okay. So in resting muscle, the myosin head cannot bind to actin, G-actin, because the binding sites have been blocked by the troponin tropomyosin complex on the F-actin. Now, F-actin is the whole actin molecule, the long molecule. This long molecule that you can see is actin, F-actin. And each one of these beads that you can see is called G-actin. So what makes up F-actin is the G-actin. So myosin cannot bind to each of those beads, those G-actins, because they've been covered, the active site have been covered by the troponin tropomyosin complex. So calcium ions which have been released upon neutral stimulation are going to be able to bind to troponin. Actually, to be specific, troponin C, and that is going to change the shape of the troponin, and is going to be able to move. Okay, so it's going to change its shape and moving tropomyosin on the F actin to expose the myosin binding sites. So what is happening is this. Look at one. Myosin heads are going to be able to hydrolyze or break down ATP, and then they become oxidized. So we are going to now have ADP because they've broken down ATP. And then myosin heads are going to be able to bind to actin. The only way they are bound to actin is because you can see these beads you can see in yellow, those are actin molecules, and then these other ones, these two you can see, these are tropomyosin. Now these other ones in different colors that you can see, those are troponin. We've got troponin I, troponin C. Okay, so the troponin C is going to be able to bind to calcium. When that happens, that will push the tropomyosin away from the active site. When the tropomyosin has been pushed out from the active site and ATP is available on myosin, the myosin can now bind to the actin and then to be able to push it behind like that. And what then when that happens, there is myosin head to be able to rotate towards the center, it's moving towards the center, this direction, towards the the center. And that is known as a power stroke. That pushing behind is known as a power stroke. When that happens, as myosin heads bind ATP, the contraction is going to detach from actin. So you need another ATP molecule for the detection to happen. So what happens if a person dies in rigor mortis? They there will not be no detaching here because the last ATP molecule has been used up. So someone who has died cannot relax because there is no ATP to make the myosin and the actin to detach. Okay. So what happens is that on the neuromuscular junction, the first thing is there was an action potential which was coming from the cell body of the nerve, and this is on the synaptic knob at the end of the nerve. So here, when the action potential reaches, synaptic vesicles are going to be released. These synaptic vesicles contain acetylcholine. Acetylcholine is going to be released. When it is released, it's going to go into the synaptic clefts and then go and bind to their receptors on the muscles. So in myasthenia gravis, what happens is that these receptors for acetylcholine are destroyed, and so even if there is acetylcholine being released, it cannot be, there is nowhere to be able to rest. So because of that, an action potential cannot be generated in the muscle. So there's going to be muscle weakness and those conditions. So when the acetylcholine comes here, what is going to happen is that as the muscle impulse is spread quickly, so the first thing is a nerve impulse triggers the release of acetylcholine from the synaptic knob into the synaptic clefts. Acetylcholine will bind to acetylcholine receptors in the motor end plate of the neuromuscular junction, and that will initiate muscle impulse in the sarcolemma of the muscle fiber. So there's going to be a muscle impulse. As the muscle impulse spreads, it's going to spread along the T tubules. Okay, spreads along the T tubules like that. And then calcium ions are going to be released from the terminal cisterna into the sarcoplasm. Look at this. Calcium, because of this action potential that has gone like this, calcium is going to be released into the sarcoplasm. Sarcoplasm is the cytoplasm. It is being released from the sarcoplasmic reticulum into the sarcoplasm. That calcium that has been released will come and bind to troponin, specifically troponin C. Troponin is going to change its shape, moving tropomyosin on the actin to expose the active site. Okay. And then myosin heads of thick filament will be able to attach to the exposed active sites, and that will cause a contraction. So you can see this was a relaxed muscle. Now here it is contracted. The length of the myosin and actin is not changing. It's just an overlap that is happening. A single axon and all the muscle fibers in contact with its branches will make up a motor unit. Individual muscle fibers will not show graded contraction. In other words, if you stimulate them now, you don't stimulate them. And then you stimulate them later, stimulate them later, stimulate them later. It will not cause them to contract. Instead, they follow the O or none principle. You should give them enough action potential necessary for them to contract. That is the threshold frequency or the threshold potential. Okay. So we've already talked about myasthenia gravis. There are different types of skeletal muscles. I've got a slow twitch and the fast twitch. The slow twitch, these ones, they are also known as slow oxidative fibers. The fast twitch, you are going to have the fast oxidative and fast glycolytic. On the slow twitch, which is slow oxidative, that's also known as type 1. These muscles are red. Why? Because they have got a high content of myoglobin. Okay, high myoglobin, they use up a lot of oxygen. So this is dependent on aerobic respiration. That's why it is oxidative. They have got low glycogen stores, meaning you like even if you have got low of the glucose that has been stored in glycogen, high lipids, they can utilize lipids, and they have got a lot of mitochondria. The mitochondria here will be used for different processes like beta oxidation. Okay, so there is oxidative phosphorylation. That's why we have got the mitochondria. For the fast twitch, these are the type 2 muscles. We have got fast oxidative, which is type 2A, fast oxidative. These ones are also red. They also depend on aerobic respiration. They have also high myoglobin. Glycogen is at least intermediate. They also have a lot of lipids. Also depend on oxidative phosphorylation. Fast glycolytic type 2B, these ones are white because they have got low mitochondria. They depend on anaerobic respiration. They highly depend on uh, they also have a high glycogen content. So meaning that because they have got high glycogen content, they'll depend on the breakdown of glycogen instead of oxidative phosphorylation for energy. So you can see the slow twitch. This one can run and do not be able to get tired because they are running slow like that. Fast twitch is at least normal, but the the fast oxidative is at least normal, but the fast glycolytic, these will be able to run very fast and they'll get to be exhausted very fast because their glycogen supply will be exhausted to be finished. Now let's talk about cardiac muscle. Unlike multinucleated skeletal muscles, each cardiac muscle cell is going to possess only one or two centrally located pale-staining nucleus. One or two. Okay. You can see how they are there, skeletal muscles. They are going to have gap junctions, and you are going to find the desmos, zones, and fascia adherents. They're also going to have gap junctions. You can see these are the spaces here. Okay, the gap junctions are there, and you also have the desmosomes. Cardiac muscles are going to have a lot of mitochondria, which are going to occupy like 40% of the cytoplasmic volume. The structure is more similar to that of skeletal muscle. They have got the thin filaments also. Thin filament is made up of three components. You have got actin, F-actin, which is made from G-actin, which bind where, which have got the myosin binding site. They also have tropomyosin. Tropomyosin is going to run along the groove of the twisted actin to be able to cover the active site so that myosin cannot be able to bind. Troponin is globular and has got three subunits: troponin I, troponin C, troponin T. When calcium binds to troponin C, a conformational change is going to occur and then remove the tropomyosin, and that is going to inhibit actin myosin interaction there. So you can see the structure of the thin filament here. This is actually actin. You can see it has got two chains. This is the thick filament, myosin. It has got two chains, the heavy chain and the light chains, which are on the head, where the active site is going to be found. And then the thin filaments, you have got three of them: actin, tropomyosin, and troponin. The actin here, this is actually F-actin. Each one of these circles you can see is a G-actin. Tropomyosin is the one which is moving along the groove to be able to cover the active site. And then the troponin, you can see C, I, and T. Okay. Some features of cardiac muscle. These muscles are involuntary. You can't control them. They are involuntary, other than the skeletal muscles which can be controlled. They also have pacemaker, which is the SA node. The SA node can generate its own action potential. So it sets the pace for the skeletal muscle. They cannot be tetanized. The meaning of tetanization is, okay, we're going to talk about the concept in front. Just like skeletal muscle, cardiac muscle are also made up of actin, tropomyosin, troponin also. The myosin is going to have the alpha heavy chain. For the cardiac muscle, the heavy chain is going to be in the atrium as the pectinate muscle of the atrium. And you also have the beta heavy chain. Those were alpha heavy chains. The beta heavy chains, these will be the trabecular carne, which are in the ventricles. Cardiac muscle has also a protein titin, which is responsible for elasticity. It attaches the myosin to the Z disc. Titin protein mutation can lead to increased stretch and dilated, okay, and dilated cardiomyopathy, because it's the one which is attaching the what is responsible for contraction to the Z disc. Gap junctions are going to be widely expressed in cardiac muscles. The protein volume which forms gap junctions is called connexins. You know that connexins, these are more circular proteins which will be able to round. They can move around themselves to close or open the gap junction so that things can be able to pass through. Synchronized contraction of cardiac muscle with help of gap junction. So these guys operate in what is known as the functional syncytium because of these same connexins. And these gap junctions are going to be localized at intercalated discs. So there's an important principle of calcium induced or calcium in, calcium released. When a small amount of calcium from the extracellular, from ECF, enters the inside of the cell, it's going to make the calcium that has been stored in the endoplasmic reticulum to be released. Cardiac muscle contraction is going to be dependent on both intracellular and extracellular calcium. So you need the calcium from outside the cell and the calcium from inside the cell to be enough to cause the calcium to push, to cause a contraction. There's also SECA, sarcoplasmic smooth endoplasmic reticulum calcium HPS pump. This pump is essential for the storage of calcium in the sarcoplasmic reticulum. You also have what is known as phospholamban. This one prevents the operation of SECA. Remember, SECA is important for the storage of calcium. So if phospholamban prevents the function of SECA, meaning that calcium will not be stored, and that will promote the contraction of muscle. So you have got things which can promote contraction of muscle, things like no epinephrine. No epinephrine is going to be able to, actually no epinephrine is going to inhibit phospholamban by making it phosphorylated, meaning to add the phosphate to phospholamban and then it will cause it to not work. And that is known as a positive ionotropic action. Norepinephrine is going to enhance calcium storage during muscle relaxation and is going to help in the upcoming contractions. I'll talk about that later. So here you have, this is your SECA pump. Phospholamban is going to prevent the work of SECA, so it prevents the storage of full calcium. Okay, let me talk about that again now. Contractility. Contractility is what we also call ionotropism. It's the ability of myocardial cells to develop force at a given muscle cell, to be able to contract, that is developing force. Agents that promote or produce an increase in contractility are known as positive iotropic agents. So the mechanism or the process is known as a positive iotropic effect. Positive iotropic agents increase the rate of tension and the peak tension, how high you can contract and how fast you do contract. Those agents which increase that are called positive ionotropic agents. And then negative will be the opposite. So mechanism of changing contractility. Contractility is going to correlate directly with the intracellular calcium concentration. And that is going to be dependent on the amount of calcium released from sarcoplasmic reticulum during excitation, contraction, coupling. If the nerves have been excited, they are going to cause the muscles to contract. So when you have got more calcium inside the cell, it causes more contraction. Okay, so they are direct proportion. The less the calcium, the less the contraction. The amount of calcium released from SR is going to depend on the size of the inward calcium current during the plateau of myocardial action potential and the amount of calcium previously stored. If you add more calcium in the sarcoplasmic reticulum, it means that you are going to be able to generate a greater action potential. All right, and also the how much calcium enters inside the cell from the outside will determine how much a contraction is going to happen. So these two, calcium entering the cell and calcium being released from the sarcoplasmic reticulum, will determine how much contraction is going to happen. When you stimulate the sympathetic nervous system and also catecholamines, these are known as positive ionotropic effects because they increase contractility. These positive ionotropic effects have three important features. They are going to increase peak tension, how much you can go in contraction, increase also the rate of tension, and also faster rate of relaxation. So they make sure that you relax fast so that you can be able to contract for a shorter period. And that will allow more time for the sarcoplasmic reticulum to be filled again to allow for another contraction. You've seen what is happening. So these are the three things. Now, let me ask, between no epinephrine and, okay, what is the major hormone produced by sympathetic nervous system? Any idea? So epinephrine, no epinephrine. Epinephrine is also called adrenaline. No epinephrine, no adrenaline. These are the hormones of the sympathetic nervous system. So these ones will promote contractility. That is positive ionotropism. Okay, these promote contractility. Now we'll talk about the excitation contraction coupling. What do we mean if we say excitation contraction coupling? As in skeletal muscles, the same way they behave, and also smooth muscles, excitation contraction coupling in cardiac muscle is going to translate action potential into the production of tension. So you first had an action potential, and then the action potential caused tension to be produced. The cardiac action potential is going to be initiated in the myocardial cell membrane, and the depolarization is going to be able to spread to the interior of the cell via the T tubules. So you first had an action potential on the an action potential. That action potential caused the release of the acetylcholine and these others, and then that acetylcholine had to bind to the receptors, and that caused a depolarization that spread via the T tubules, and that is what led to the release of calcium from Seca. So you can see that you are going to have on the T tubules, you have got the L-type calcium channels, which are known as the dihydropyridine receptors. Okay, so from the dihydropyridine receptors, these are going to be able to allow calcium from extracellular fluid to enter the intracellular fluid. So you first have a cardiac action potential, which can be generated by the pacemaker, and then calcium is going to enter the cells during the plateau phase. If you look on the action potential for a cardiac muscle action potential diagram, when calcium enters, the calcium that has been has entered the cell is going to cause calcium to be released from the cell, and then calcium is going to be able to bind to troponin C. When calcium binds to troponin C, it means that tropomyosin is going to move from actin, and then cross bridge will be formed, and that is going to result into tension. If you have got ATP, calcium can actually go back into sarcoplasmic reticulum. The myosin will be able to move from the actin. That will be able to cause a relaxation. So we also have the length tension relationship here. That states that changes in length changes in length are going to be associated with diastolic volume changes. Meaning that if the heart receives blood, it's going to contract. There's going to be a change in length. Tension changes are associated with pressure changes. So if there's a change in pressure in the heart, then there's going to be a change in tension. So you can see diastolic volume and pressure. When the volume increases, the pressure also be increasing in the heart. There will be a normal physiological limit such that when you continue to increase the volume, pressure will no longer increase in the heart to start decreasing. So increase in volume is going to lead to increase in pressure within physiological limits, and that is known as Frank-Starling's law. The most common injury that cardiac muscles can be able to suffer is ischemia, the death or the damage of tissue because there is no oxygen supply when the coronary arteries have been occluded or they've been blocked. Okay, because there are no satellite muscles in the cardiac muscle have got limited potential to regenerate. So if there's a problem with the coronary circulation, the heart tissue will be able to die, and they do not regenerate easily. So a person is more likely not to survive. Now talk about smooth muscle. Smooth muscle is specialized for slow, steady contractions and is going to be controlled by a variety of involuntary mechanisms. Instead of having the T tubules, you are going to have curvature in smooth muscle. You also don't have Z discs, instead you have got it dense bodies where the myosin and the actin are going to be able to bind. So the cavernous of smooth muscle will contain several pumps and ion channels which serve to organize protein signaling calcium release at myofibrils. The characteristic contractile activity of smooth muscle is generated by myofibrillar arrays of actin. In smooth cells, what will happen is that a bundle of thin and thick myofilaments, they will be able to cross obliquely across the cell. Myosin filaments have a less regular arrangement among the thin filaments, different from skeletal and cardiac muscles. The actin filaments also don't have troponin. Instead of having troponin, they are going to have calmodulin. Apart from that, they also have calcium sensitive myosin light chain kinases. Remember the troponin is where calcium was binding. Because the troponin is not there, you have got calmodulin. You also have sensitive myosin light chain kinase. Smooth muscle cells have an elaborate array of desmins. You have got other proteins known as desmins, dense bodies, already talked about that. These are the calvera that we are mentioning, these depressions here. Smooth muscles also have pacemakers, which are known as interstitial cells of Kajal. They don't have ZDs, as we've said. They also don't have titin, meaning that they are not elastic. Okay. They don't have troponin C, meaning that calcium binding protein, what binds calcium is going to become calmodulin because it's what replaces troponin C. So because they do not have titin, they can continue to stress, and that is to stretch. That is known as plasticity. They can go on stretching and stretching and stretching. They will not be able to go to the original shape. Example, the uterus can actually stress and stretch and be like that for a long period of time. That is why for 9 months your uterus is still stretched. Because if it had that property of elasticity, it means that if you pull it, it will go back to its original position. So if it goes back to its original position, imagine you are pregnant, that would exert a greater pressure on the child. Okay, also the blood. Imagine if it could go back to its original position and someone has got training in the blood, what would happen is that they couldn't control that. Prolonged state of contraction with little use of ATP, and that is known as the latch bridge phenomenon. Because normally, for when you contract very much, you're supposed to be having ATP to continue like the contraction like that. Because with time, tissue will be able to die, and then there's going to be that relaxation. But the latch bridge phenomenon is telling us that these smooth muscles can contract for long without use of ATP. There are different types of smooth muscles. I've got a single unit or unitary smooth muscles. These ones are the common ones, the most common, and they have got gap junctions. It is important to understand that they have got gap junction because the gap junctions make them to work as a sensation, to work together. They also exhibit pacemaker activity, which is modulated by hormones. They have a degree of electrical coupling between cells, and therefore they are going to permit coordinate contraction of the organ. Where are these going to be found? They're going to be present in the GIT, so that when there's contraction at one point, there's going to be contraction on another point, and just as that. Also in the urinary bladder, uterus, also ureter. Mouth unit smooth muscles do not have gap junctions. They are present in the eye, the iris, ciliary muscle of the lens, and also the vas difference. You also have vascular smooth muscles. From the word vascular, they are found in blood vessels, the arterioles. Now, the excitation contraction coupling of the unitary smooth muscles. Also, for smooth muscle, what will happen is that first, there's going to be opening of calcium channels in the plasma membrane, and that will lead to calcium entering into the inside of the cell. And 80% of the calcium needed in smooth muscle contraction is going to enter the cell from the extracellular fluid, while 20% is going to be released from the sarcoplasmic reticulum. as a calcium induced, calcium released. Calcium in the cytoplasm is going to be able to bind to the regulatory protein calmodulin to form calcium calmodulin complex. And then calmodulin by itself cannot activate myosin light chain kinase. So what will happen is that calcium calmodulin complex, when calcium binds to calmodulin, then myosin light chain kinase is going to change its state from being inactive to being active. Now, this is a kinase. After it has been activated, it is going to be able to activate myosin ATPase, okay, by phosphorylating the myosin heads. So, activated myosin light chain kinase will activate myosin ATPase. The ATPase adds a phosphate. So, to add a phosphate to the head of the myosin, and the myosin ATPase will become active only when phosphorylated by myosin light chain. And what happens is that now the myosin ATPase can initiate the cross bridge cycle. Once the myosin ATPase is activated, the cross bridge cycle is very similar to that seen in skeletal muscle or striated muscle. And then there's going to be muscle relaxation later in a multiple step process. Okay, cytoplasmic calcium concentration is going to be reduced back to normal through the activity of sarcoplasmic and plasmic membrane calcium ATPase pump. Apart from that, you also have plasma membrane sodium calcium exchanger, which will be removing calcium from the cytoplasm while sodium is entering. When there's a reduction of calcium concentration, that is going to lead to dissociation, which is the release of calcium from calmodulin, and inactivation of the myosin light chain kinase is going to happen. Myosin ATPase will become again inactive when the phosphate group is removed by myosin light chain phosphatase. Remember, the myosin light chain kinase was adding a phosphate, phosphatase is removing the phosphate. After the myosin ATPase is dephosphorylated, ATPase activity again is going to be inhibited and the cross bridge cycles becomes hot. They will stop. Okay. So that is an overview of the muscle physiology. Which of the following is a characteristic of white muscle? It is responsible for slower muscle movement. It has a high mitochondrial content. Primarily utilizes aerobic metabolism. It has a greater mass per motor unit. It contains high amount of myoglobin. So white muscle do not contain high amount of myoglobin. That's not true. And it doesn't utilize anaerobic respiration. It's not aerobic but anaerobic. So it's going to have no high content of mitochondria, because high content of mitochondria is supposed to be for the red muscles, responsible for slower muscle movement. It's not for slower because these are fast muscles. Okay, so the best one, so you can pick it as a greater mass per motor unit. For other questions, we'll be able to answer in the other video where we'll just talk about questions.